Okay, so if everyone can hear me, then I think I have to thank Faith for the opportunity. I think it is an honor. It's an honor to actually be here today to give you this webinar on COVID-19 under the auspice of IUIS. So I'm Lisa Ng from the Singapore Immunology Network, A-Star from Singapore. And I have been actually working on viral infections, specifically focusing on infection and immunity for many years now. And my area of interest is really on arboviral infections. But I've also, like most of you out there around the world, you have also all jumped into the bandwagon for COVID-19 since the pandemic has started this year. Just to give you a little bit of background, because we couldn't hear Faith, my background many years ago during my PhD days was actually on coronaviruses, and that was actually pre-SARS days, the SARS 2003 outbreak. So it seems to have keep coming and going into my life, and I think each episode has been really, really interesting and exciting. So what I'm going to tell you today, it's about my experiences together with the rest of the research and clinical community here in Singapore on COVID-19. So let me move to the next slide. I know that many of you are from all over the world, so just to give you a little bit of introduction, or rather a little, just to tell you where Singapore is. So we are in Southeast Asia. We are a city-state in Southeast Asia, just south of Malaysia and north of Indonesia, and this is the Philippines, and China is here up north, and this is the rest of Southeast Asia. So you could see we are a key travel hub between Europe and Australia. So in fact, we have many passengers who typically stop by in Singapore at the Singapore Changi Airport, which I believe some of you are familiar. For those of you who have not, well, I hope that maybe when we have better days in 2021 onwards, please come down to Singapore and you will see how beautiful the country is. So you could see that unlike many countries, I believe that you have, when you follow the news, in end December, China released a statement indicating of some strange going ons that was known as the Wuhan coronavirus. And so obviously the rest of January 2020, I think the whole world was actually put on high alert. So in fact, for Singapore, we had the first known case as early as on the 22nd of January. That is just at the beginning of the lunar Chinese New Year. And this data was obtained on June 14 yesterday. Even until now, we still have new cases being reported. And clearly, we have 26 deaths. I think the clinical community have been managing very well. We have actually quite a few we have more than 40,000 cases that are confirmed, of which 220 are in the hospitals in Singapore, as you see later. And we have more than 10,000 which are in isolation. So these are typically people who were suspect. So this is how it is. So when this started, obviously the Singapore government was very, very cautious. And then I believe that if you have been following the news, you could see that the controls were very, very strict. Unfortunately, like all over the world, I think the cases were not really decreasing. So by early April, the government decided to what we call the circuit breaker. It's not a full lockdown, but rather a circuit breaker. So you could see this is how it looks like. You know, typically this is an area in downtown, right near the Singapore River. It's normally filled with tourists and people working in the financial district. And this was taken when I was actually doing my run. We are still allowed to go out for exercise, you know, and you could see this totally empty. And this is also the Singapore River in another part of a very busy Singapore. So typically the circuit breaker was supposed to end on 4th May, but you know, by the end of April, we realized that the cases were not reducing, so it was extended. And it was till 2nd June, and so Singapore just started out, we were just moving out of the phase 1 of the circuit breaker, and so life is not totally back to normal yet. Retail shops are still closed. But just to show you a little bit of data here, you could see from here that since the circuit breaker was imposed, and you could see that actually the number of cases in the community was actually was reduced sharply. And of course, what has been causing the increase even up to this day was actually the foreign workers in the dormitories that actually had the spread, and this is still ongoing at this point, but it's, again, like I said, it's under control. So in terms of COVID-19 studies in Singapore, so this is data was actually obtained from our collaborators at the National Center of Infectious Diseases, and which is the study called PROTECT, and this is a multi-agency study for all COVID-19 patients that involves both the clinical community and also the research community like myself. So I just have to really acknowledge with how the entire patient cohort that you're going to be hearing about today is really led by the 3 key people here, right? Typically, mainly Professor Yi-Sin Liu. She's the Executive Director of the National Center the National Center of Infectious Diseases, known as NCID, and also Professor David Lai, whom I'm sure some of you must have seen him on TV quite a little bit on the various treatments of COVID-19, and also Dr. Barnaby Yang, who really very instrumental in pulling the whole cohort together that involves several hospitals in Singapore and also with the different research partners. So what you're going to hear about is really the patient cohorts that have been collected and the various kind of studies that were done. And I'm just going to be showing you a little bit because if you were to read about COVID-19 all around the world, there's always the description where there are some cases where some patients actually get very severe disease, and most patients are fairly mild, and then there's also some that are fairly moderate. So the patients, I mean, Singapore is not really unique, just like anywhere else in the world. The patients were also, we have different categories. We have the mild ones, which has no pneumonia, which is just the fever and dry cough, as you'll see later. And we do have the moderate, where patients actually have pneumonia without hypoxia, and the more severe ones. And obviously, a subset of these will then be in the intensive care unit. So remember these 3 classifications, because later on, you'll see that the various projects that we do to understand the immune profiling, from cytokines, from serology to even a neutralization and antibodies, these 3 categories prove to be very useful in our data analysis. So over at the Singapore Immunology Network, we have been having a long-standing collaboration with the NCID for several years on many different outbreaks. We could keep to this discussion probably another time. So led by Professor Laurent Rainier, which some of you are acquainted with. So we have the SARS immune monitoring platform that you could see that it's effectively 7 core technology platforms that will actually feed into a lot of the research questions that we do, that will enable us to look at immune profiling from cells to plasma to other clinical specimens. So what we typically do, it's really from the clinics, we will then actually look at deep immune phenotyping, and all of this, we will actually try to develop, to really understand the mechanisms of infection and inflammation, and how some of this information will then be used to feed into areas such as to identify specific biomarkers that could even be used as a prognosis, and even as a point of care tests, and so on and so forth. So, okay, just a little bit of introduction in case you are interested. Well, this COVID-19 was actually caused by a very huge coronavirus. Okay, so it's what we call the coronavirus 2 or SARS-CoV-2, well, because there's SARS-CoV-1 back in 2003. Now, so it belongs to the Coronaviridae family of virus. And you could see from here that the genome size is really huge. It's about 30 kilobase big. Compared to many other smaller RNA viruses like flavivirus or even alpha viruses, they are about 10 kb large. So you can see that this is 3 times. So which means that, the complexity of even studying the pathogen itself is really a lot more challenging compared to many other RNA viruses. Okay. So I think some of you are familiar. There's the usual nucleocapsid protein, there's the envelope, and then there's a membrane, and the famous or infamous spike protein, which it proves to be a target for not just diagnostics, but also in terms of various treatment strategies. Okay, so what about the typical symptoms of COVID-19? I mean, if I were to explain fever, dry cough, and shortness of breath, or even a sore throat and headache, I think this is fairly similar to many other viral infections. Even the flu, or also some of the other viral infections. So you could see from here that sometimes it's not so easy to actually discriminate, especially during the early phase. But I guess what is quite unique is actually the dry cough and the shortness of breath. But still, there's a lot of overlaps between this and also the other infections. So, which is why first-line diagnostics, it's paramount. Okay, so I think all of us are aware that the PCR, or what we call the RT-PCR molecular diagnostics, remain somewhat as a gold standard, for early detection. And this is when you will be able to detect the viral RNA in the clinical specimen, specifically the nasal swabs. In this case, not so much in the blood, but we do know. We are also aware that the window period for molecular diagnostics is also quite low. You see, it's very, very short, it's very limited. And, the moment it goes beyond 2 weeks, sometimes you are not able to detect them anymore. And this is when you need other kind of detection systems, in order to complement the early phase. And this is where some of the serological assays will be useful. Now, there still remains a big challenge, to use serological assays to detect the antigen. And this, I think the sensitivity, it's always a challenge. But then, the other kind of assays that if you want to talk about exposure to the infection, then we'll be talking about the IgGs, or the IgMs, or even the IgAs, which I think if you follow the literature, there are reports where you can actually be used. So, in order to actually improve the entire spectrum of detection, you can't just rely on one type of assays. Even you have a few, the platforms could be different, and this very often it could differ from the different sensitivity and also specificity. As you remember, this is a very huge virus, which sometimes can also be a little complicated. So let's coming on to the first part of our efforts to actually develop good detection reagents. In this case, serological assays to detect the antibody profiles. And how some of this could then be used to funnel it into detection assays. So I would really like to acknowledge the various people in this work. You could see that it's a lot of work, and until now, although we have completed a few series of studies, they are still screening, because we just have patients that are coming in, and our approach is not so much on the recombinant protein, but is what we call the epitope mapping. So in this next 10 minutes, 5 to 10 minutes, it's actually, you'll see that there are 2 different stories, all right, that is embedded into this extended study of antibody profiling. So the first is actually led by Nakia and Cheryl in the lab, and of course, and the rest of the other members. And the second story, it's actually led by 2 very brilliant senior postdocs. Well, they are going to be to have the lab someday, very soon, I believe, which is Chek Ming and Guillaume. And obviously, we have other collaborators, Matthew and Zoe from other labs, and also the other researchers and PhD students that really play a big role in this whole effort. So what did they do? So as I mentioned, our approach is slightly different from what is out there. It is an ELISA-based assay, but we are not using the whole antigen, but rather it is specific short peptides that are specific. So in this case, what they had done was, we had a library of linear peptides overlapping by about 10 mers, and by 10 amino acids. And you could see from here the first screen by using the patient's plasma or serum from the COVID-19 patients. You could see from here that specific pools actually lit up. So obviously, most of them were in the spike protein, right? We have quite a few at the S14 and the S20 and the S21, but we also have a pool, a set of peptides that actually lit up in the nucleocapsid. And when we then carried on to further identify the specific epitopes, you could see from here that it's not every peptide that actually was positive. In fact, for the different pools, you could see that it's one unique region. Okay, so obviously this is interesting because clearly for the pool of S20, you can see that it's a series. So which means that this particular region is fairly antigenic. And so it's also important to ensure that the specificity it's there. So we also have the similar peptide from corresponding peptide from our SARS library, the SARS-CoV, the one in 2003. And when we did, we actually performed the peptide-based ELISA of the same patient's materials against the corresponding SARS-CoV epitope. You could see that actually some do not recognize, while there's some levels of cross reactivity here. And if you were to look at the localization of where these epitopes are, well, you could see that actually all of them are located on the surface of the viral protein, except for the S20P2, which is actually partially buried in the 3D structure. Okay, so and then this is the one in the nucleocapsid protein. Now, so that was actually done on a small cohort of patients. Now, what happens if these epitopes were then used to screen in a peptide-based ELISA against a larger cohort? As I said, we had patients coming in, and then some of the patients, we followed them longitudinally. Well, you could see from here, result speaks for itself. 4 out of the epitopes actually performed very well, right? Obviously, the S6 was not so perfect, but then you see for the S14, the S21, even the S20 and the N4, you could see that it's very, very specific. And I'd just like to point out to your attention, as I said, we actually had our outbreak of SARS back in 2003. So we actually invited some of these recovered SARS patients back to the study, and then we collected fresh blood from there, and then we used them as a control. It will be interesting to see if this could actually recognize the SARS-CoV-2 antigens. And you could see that actually most are clean, right? We also have our healthy controls, with the exception of a few, 1 or 2 patients that still could, that had low levels of antibodies that could cross-react with the SARS-CoV-2. Now, what we then did later on was actually to make use of a statistical analysis, what we call the Uden-J statistical method, which is a method that provides an optimal cutoff value for each of the peptide. And because of this value, it will allow one to determine the sensitivity and also the specificity profiles of the assay in one single test. So what we done here with the help of a bioinformatician was actually to predict, so if we were to use them in different combinations, are we going to bring the detection rate to 100%, without compromising the specificity. So you could see from here that, indeed, there were some epitopes that would actually, increase the detection rate to 100%. So actually this gives a potential idea that, some of these epitopes could be actually translated onto to different kind of a detection platform. Either is it an immunoblot, or is it beads, or even a lateral flow of sorts. So it's really there's a lot of various options and the different things that we could actually do from here. So this will really enable the development downstream. But I have to say, the discovery element of it remains of prime importance, because without that, we were not able to actually understand any of this. Now, if you remember the classification of the patients in Singapore, as I showed earlier, we had 3 types. We had the different types of the 3 categories of disease severity. We have the mild, which are those patients that without any pneumonia. We have the moderate, which is what we call patients with pneumonia without hypoxia. And then we had the most severe group where the patients had pneumonia and hypoxia. So using these epitopes, you could even see that we could even differentiate them quite nicely here. So the association of the antibody responses, it actually correlates quite nicely with clinical severity. So I believe different cohorts from around the world also have shown this by using recombinant system, or even some with the neutralization assays. So again, it has 2 elements that will be very useful. Now, we had also, because of with the help of our clinical partners, Barnaby and friends, they also enable us to re-separate to reclassify the patients into different types. We have those patients that, like I said, with pneumonia, and then those that had went into ICU, and those that needed additional ventilator. So what is interesting is that the different epitopes actually have a slightly different profile, okay? So efforts are ongoing now to actually further characterize some of these epitopes. Okay, so I believe, like I said, many of you are probably in the same situation as Singapore, which is biosafety, right? So, unlike other arboviruses like dengue, maybe even chikungunya, which is we could handle it under a BSL-2 condition. Now, SARS-CoV-2, it's really a strictly labeled BSL-3 pathogen here in Singapore, and I think for many countries as well. So as a result, actually this limits the types of studies and even experiments that one could do. I mean, it's very different to be working in a BSL-2 condition compared to when you had to handle this in a BSL-3 condition. So, which is what I'm going to, the point which I believe where I'm heading to, it's the handling of live viruses, right? So which means it's not possible for BSL-2. So, we were this is the work that was done in collaboration with our collaborators at the Defense Science Organization, because they have the BSL-3 facilities. So, they actually kindly perform a live virus neutralization from our patient's serum, in this case, and to actually establish if these patients actually contain neutralization antibodies, and indeed they do. But also in SIGN, by the lab led by Cheng Yi Wang, his team actually developed this BSL-2 lentiviral pseudotype SARS-CoV-2 assay, which is based on the S glycoprotein. And so that one, we could handle it in BSL-2. And you could see from here, when we compare the 2 different assays, right, the pseudotype virus and the live virus, you could see that the IC50 values are fairly close. Well, you'll never get 100%, but at least you show that this is actually fairly stable and quite reliable, to actually to assess if the patient's antibodies have neutralizing antibodies. So with this, we then expanded the neutralization. This is the work of the scientist, the senior scientist in Cheng Yi's lab, Bei. So you could see that almost all the patients here actually could neutralize in a dose-dependent level. So this actually reconfirms that these patients carry the neutralization antibodies. Now we're going to put the different work together. If you remember the epitopes I've shown earlier, so if you could see from here, this is what we have, from the same set of patients. Well, there are, of course, some patients are exclusive, but most of them are overlapping. And you could see from here that we went on to further characterize two key epitopes, which is the S14 and the S21. Now, what is so special about this? Why these two? So if you were to look specifically at the localization, right? So you can see that the S14 is actually localized in close proximity to the receptor binding domain, what we call the RBD, while the S21P2 is actually covers part of the fusion peptide, which is known to be a somewhat important area, a region in our virus replication for coronaviruses, right? So we went on to further study these two epitopes, and moreover, with the neutralization assay that we have shown earlier, you could see from here that the antibodies directed against these two epitopes actually sort of associate very nicely with the neutralization. So does this mean that these antibodies could, be neutralizing? So obviously that's another study, but for this, to finish up this piece of the work, so Guillaume and Cheng-Ming, they actually did a very painful experiment where they actually tried to deplete away the specific S14 and the S21 part, with a depletion assay. Then you could see from here, when they remeasured the titers this is pre-depletion and after depletion, you could see that it actually reduced the detection. And then, so it means that the depletion assay was successful. And of course, this is a control experiment where they did the cross ELISA, right? So, for depleted S21, they actually ran it on the S14 and vice versa. And after that, from the depleted serum, they actually then carried out the in vitro neutralization. And you could see from here, after the depletion, the capacity to neutralize SARS-CoV-2 was actually reduced by 20%. So obviously, we will not be expecting 100% because there will be other regions across the S viral glycoprotein that are also neutralizing. But just by these two epitopes alone, you could see from here that it carries about 20% of the neutralizing capacity. So I think there's a lot of work that will keep the team busy for the next 6 months to a year to really do the further characterization of some of these work. So I'll just move on to the second part of my talk, which is the next 5 minutes or so, where it's really to further understand some of the other elements of the immunopathogenesis of COVID-2, right? So this is a review that was written by a postdoc, Matthew, in the lab of Laurent Rainier, and also with Cheng-Ming from the lab. And this just shows a very nice path, what happens when the virus infects the cells, and then a whole load of cytokines, or what we call immune mediators, are actually produced, and then this then causes the inflammation. And the next part of the work actually involved many other people, also 2 other senior PIs, that really played a very big role in the data analysis and also the panel. So it's also led by CY Yihao, and also with two very, very dedicated researchers of the platform. So what we showed here was that, the profile, the cytokines profile of the COVID-19 patients are very different. Okay, so remember again, there are the three categories: the no pneumonia, pneumonia without hypoxia, and with hypoxia. You could see from here that the pro-inflammatory signatures are also quite different. You can see that it gets more intense as the patients get more severe. And when we did actually network analysis, data mining of all the cytokines that have been published out there, and you could see from here that actually there's a very common group of immune mediators that are actually that we found between the influenza infection and the SARS-CoV-2. So I think, again, you see the IP10 has a big role here. Now, the next thing that I would just like to show is that, as I mentioned, that we do have some patients that were in the intensive care unit, and I'd just like to highlight that we found specifically two markers that were very unique. When the patients were almost recovering, those in the ICU, their levels actually returned back to normal. So one is the HGF and the other one is a VEGF. So potentially, this could mean that, this could serve as an early indicator of poor prognosis, and potentially it may help to guide, preemptive clinical decisions, right? So to move on, based on this entire series of cytokine studies, we also had more data from the clinics, and then from a subset of the big set of the SG 100 patients, we then zoom into a smaller set of patients. Why so? Because we found that potentially out of that whole series of signatures, we found two markers, which is IP10 and IL-6, that could stratify the disease severity even further. So you could see from here that these two markers could actually be used to distinguish patients with or without pneumonia and also hypoxia. So, so everyone, is always looking at the disease severity, but there's another thing that CY and Yihao then went on to look was, what about recovery? Are there any markers that, at the point when the patient has been hospitalized, the first point when they were first admitted, could their levels of mediators be used to estimate how soon they will recover? And again, the same two markers, IP10 and IL-6, actually showed that, patients who actually are more likely to recover within the week, right, with less than 7 days, compared to those who did not, again, this could be used to differentiate the patients. So, so this is where we are now. We have several ongoing studies, to look at the subsets of the different immune cells that were known to produce some of these markers. And also we are really trying to understand, in terms of recovery, how some of these patients are coping and things like that. So, with all this, it just allows us to actually not just for diagnostics, but also as for prognostics, and that could be used to further translate into different types of point of care tests for the hospitals that, again, that will be very useful to help relieve the burden of the healthcare. So I'll just like to end off quickly. Really, as I said, it is the work of many institutions, right? Of course, those at the Singapore Immunology Network, led by the various PIs, for the different work that you have seen, and also our clinical partners at the NCID, and also the rest of the clinicians who were very much part of the PROTECT project here, you could see, and also our partners at Duke NUS, specifically Danny Anderson and the team, that they were very instrumental to help us some of the inactivation of the samples, and also our friends at the Defense Science Organization. So yes, and just hope that, again, this is a picture, shared by Professor Liu from her center, and you will see these are all the healthcare workers who are still working hard, still taking care of our patients to ensure that, they recover quickly. And this is the most recent picture, like I said, even though we are out of the circuit breaker, the place is still empty. So I just hope that, as we continue, there will be clear skies ahead, so that we could just start to travel again, which is something I really miss, Faith. You know, I really miss traveling so that I could continue to go places and try out new and good food and good wines. So with that, I will thank you for your attention and happy to take any questions now. Thank you.
Thanks, Lisa. Thank you. I'm not sure you can hear me now, Lisa. Can you hear me?
Yes, 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 yes. Yes, I think everybody
wonderful.
can.
So it always happens when I'm doing the webinar that the sound goes, but when other moderators do it, it sounds fine. So it'll be all right. It'll be all right. But thank you for a great webinar. So there's a few questions. I'd like to encourage everyone to ask more questions. There are not so many today. But Lisa, maybe you could just start by explaining how come the spread in Singapore is generally limited compared to other places that we've seen.
You're talking about the cases?
The spread of COVID-19, yeah.
Yeah, well, because people take the rules very seriously here. So, you know, the correct answer is that there are rules, and when rules are implemented, you follow them. So, no, I mean, the fact that I think the sense of awareness is high because we have gone through the SARS episode back in 2003. So, you know, there's this awareness that it should not be taken lightly. I mean, all respiratory infections should never be taken lightly. So there's a lot of awareness early on. So when this thing clearly this was out in the open, I think many people were very cautious, and they really took, you know, the safe distancing measures were really put in place. So it is very unfortunate that the dormitories were, you know, created some kind of a complication, but like I said, that is also under control, and we just have to let it, you know, time. We need time, yeah.
Okay, and Lisa, from your experience with coronaviruses that you've looked at before, should we expect that there will be long-term damage in children, in adults? Will there be long-term damage, and will this be different in those two groups?
Yeah, I think this is a very good question, and I think it's still very unclear. I think especially the effects of a SARS-CoV-2 in children is still very unclear. Although there have been reports to say that typically they are mild, but then we are also aware that, you know, in some countries there also the cases of Kawasaki has also increased, and that is actually very severe. So I think that is still very unknown. So is it to do with host genetics? Perhaps, right? So I think in terms of long-term damage, then it really also depends on how severe. I think right now we don't know enough. For SARS, you know, people recover. For those who recover, they seem to recover. But for SARS-CoV, I think we have been reading quite a few interesting or conflicting reports from places that they seem to have revert and things like that. So I think we still do not know enough, and that's why it's also quite worrying. Yeah.
Okay. The other question has been on asymptomatic carriers. Whether you've done any work on those and what their immune responses look like, whether they're reservoirs and transmitting. So there's also been lots of up and down about that.
Yeah, indeed. I think this is where an area that is still unknown, and we are looking at it. So with the NCID clinicians, that's what we are looking at. And so for Singapore, clearly they have to be called asymptomatic COVID-19 if they are PCR positive. So we do see that in some of these cases they are PCR positive, but then obviously they are symptomless. So it's still quite unclear. Yeah.
So you haven't done any immunology on them yet?
We are doing now. We are.
Okay,
it's ongoing. It's ongoing. Yeah.
Okay. The other questions I've had are about the levels of IL-6. Someone has commented that the range is so wide. So they're not sure how we can use this as a marker when we've got such a wide range. Yeah.
I think that is a very good question, and the person who raised that question, you have very sharp eyes. As you know, the detection of IL-6 for Luminex is not the best, right? And also for the different ELISA bead assays, you can see that it's also quite varied. So clearly, for patients that have high levels, that's not a problem. We detect it very clearly. But then for those patients that have the slightly, the levels are not exceptionally high, then they are on the low end, then that could compromise. So which is why we are also, constantly trying to improve the detection, the sensitivity, and so we actually move on to some other assays that are more sensitive. And you could see that the results could differ so much, but the trends are even more spectacular.
Yeah, and just related to that, when you look at the IL-6 or IP10 levels from day 1 to day 10, so when you look at the difference in the levels, is that a better marker than the one-time measurement?
We have been looking, we followed them, we have the longitudinal. So you could see from here that the data that I showed, the date where they were the first time point where before they show the onset, of the severe symptoms. So just before, so we have before, we have after, and this is the point where before we see that they manifest into something more serious. So it is so if the assays are the detection limits improve for I think IP10 is good, but if the detection limits are further improved or enhanced for IL-6, I think it will change the way we look at clinical management.
Yeah, okay, thank you. There have also been some questions about conformational epitopes.
Yes.
And the fact that you use the peptide arrays. Obviously, then you showed us some data on some functional data, but there are several people asking about what your thoughts are on potentially on conformational epitopes.
Yeah, definitely they are around. And as I said, that two linear, the S14 and the S21 are two linear epitopes that were found that were clearly specific and detected at high levels. And when we did the depletion, we found that the neutralization capacity was reduced by 20%. So which means that this leaves another 80%. So what are the epitopes? So it's going to be a mix of conformational and perhaps other epitopes, other linear epitopes that we have not identified, or the rest could be a mix of conformational. So definitely they are. But it's just that our system, we were focusing, this approach was the linear B-cell epitope approach. Yeah, so keep hunting. I'm sure you will find that. Yeah.
For sure. And another question is, when you have your ELISA data had really good specificity and sensitivity, but once patients recover, how long do the antibodies? How far have you looked to see how long they have?
Yeah. So we are still looking. So we are into month 3 and 4 of the earlier batch. So we are collecting the data. So I think potentially there are, it's not as long lasting as the other arboviral infections. I think this is quite known for coronavirus. So I think we need more data, and we really need other cohorts to really compare this.
Yes.
So I think for those of you from different parts of the world, if you're interested in some of these epitopes, please drop me a line. We are very happy to share some of the available reagents that we have, because it's important to get as much data as you can. And if this is going to be useful for your cohort, we will be most glad to share them.
I have a question also on the N protein.
Yes.
Is this good for diagnostics?
It is. You could see that it is very, very sensitive, and also specific. But it should not be used alone. We will probably in combination with the other S as well. So we do know that for some of the IgG assays, people are using S antigen and also the N antigen. But I think to just fix on one might not be good enough. I think it will be good to have two types, because on our end, our data, we found that the earlier question where what about patients that much later on in the disease, so we can see their profile. So I can tell us that the profile is slightly different.
Okay, and have you found that any antibodies against the N protein can neutralize?
We are doing that at the moment.
Okay. The other question is about that some studies have shown that IgG antibodies are appearing before IgM, which is the reverse of what we understand from immunology. Do these share the same epitopes with other coronaviruses? I think you showed us a little bit of cross-reactivity at the beginning. So that's one question. And then any role for memory T cells?
Yes. Okay. So the role of memory T cells, we are doing it. It's a separate study, so that is ongoing at this point. Also, there are other groups that are also reporting on T cells. I think you can find it out there. So maybe, but of course, the cohorts will be different. So back to your first question, which is about the IgM and IgG.
Yes.
Now, I did not show any data on IgM for today. Because the system, we are still refining it for IgM, but I can tell you that actually potentially the epitopes are different.
Okay.
And to the question where does the IgG come earlier than IgM, I think it's still very debatable at this stage. I think it also depends on what system you are using and also how sensitive and how specific. I think this is where there's a lot of issues with the various type of assays. I think it's still the challenge to catch the right time. I think it's because there's going to be and also depends on, you might collect the sample from this particular patient, but only if the patient shows a lot of symptoms, and they sort of could count the date back or when was the first onset. But there could be some patients that probably they were not aware, they could actually be harboring the infection longer than they knew, and then they said, oh, I only started feeling unwell yesterday. But so again, this variation is going to be quite broad, and this also will complicate the type of assays to be used. Yeah. So it still remains a challenge. Yeah.
Okay. There's a lot of questions coming now. Someone's asking whether we are now clear on the total incubation period for COVID-19, and I think related to that is, are you seeing any mutations in the genome with your viruses in Singapore?
Yes, there are. So this, if you were to go to GIS8, so the Singapore representative of GIS8 is Dr. Sebastian Mauerstro. I think he has been loading the sequences and comparing with all the sequences that are found. So if you go into GIS8, you will see, there are different clades that are evolving. So I think that's an interesting thing. So yes, just like any other country, Singapore is not unique. There are mutations, and it's happening. Yeah.
Okay. There's another question similar to what we've done before. There's one on T memory cells.
Yeah.
That can we think of a COVID vaccine that can modulate T memory cells in the lungs and in the tracheal airways, CD8 T cells? I think you focus mostly on serology, yeah?
Serology, and I said we are looking at T cells from the cells as well, so that is ongoing. We have another project, so stay tuned. I hope this is the first talk of all. We have several studies. As I said, it's a multinational effort, so we have many other studies that are upcoming. Yeah.
Okay.
So yes, it's being done, but as for the answer to the vaccine, I think that is one of the strategies as well. So there are also projects that are looking at that.
Okay.
Yeah, not led by me, but some of my colleagues. Yeah.
Okay. And do you think some of the peptides you show us could be potential vaccine candidates?
I very much wish so. I'm sure. So any companies out there, we are open. I'm happy to discuss with you.
Yeah. Okay. And is Singapore involved in any plasma therapy studies?
That has been discussed. I think without the availability of a monoclonal antibody, this is one of the approach. Until something comes along. So I think just like say Singapore is not unique in terms of this. So it is being discussed.
Okay. Do you see any evidence that antibodies might enhance infections like we see in dengue? In SARS patients, and how do you plan to avoid this in vaccine development?
So that I think the concept of ADE has always been discussed. I think the moment you have neutralizing antibodies, then the next question is what happens when you have sub-neutralizing antibodies, and that's where the whole idea of ADE. So it's possible. We have not done the experiment, but it's possible. But again, this then leads to the earlier point that I mentioned, that unlike dengue or some other arboviral infections, the antibody, the IgG seems to be not as long lasting as what we see out there. So maybe that potentially that might lower the the chances, but that also means that, how useful will a vaccine be. But then I think the vaccine is still the way to go at this point. So I'm not sure if I answered your question directly, but I said it's definitely possible. We have not done it, and we don't know. Yeah.
Okay. And I mean, it's also just related to that. If you did have vaccines, of course, there's so much debate on how long the protection will last.
Yes.
But, I think that's a question that's tough for anyone.
Yeah. I mean, then it could be something like the flu vaccines. Then there will be an update every year or things like that. And then of course, then that is if SARS or SARS-CoV, if there's going to be a SARS-CoV-3, touch wood, I don't know if I mean, but the thing is that then it really depends what might happen in the subsequent years. So you can just see, I mean, the first time we had the SARS was 17 years ago, and now, so is it going to come again 20 years down the road? We don't know.
Yes.
Yeah.
Okay, just working through. Someone asked before whether you had any cell types that were predictive of recovery. What markers, prognostic, good outcome?
Yes. We have done that, and the paper is currently under revision. Not so much for recovery, but more prognostic. So, of course, that also depends on how early on you actually collect the blood from here. So, potentially, there are some reports out there that show that neutrophils that could be potentially be useful. But then, of course, there are different subclasses of neutrophils. So I think that's where it'll be quite interesting to see how it looks, in the coming weeks and coming months.
Okay.
So yes, it's possible. Yeah.
Great. Okay, so I think we've covered most of the ones. I mean, there's one from India asking if you can comment on the lower fatality rate. In India. What do you think? Is it genetics, immunity? Is it the strain of the virus? It wasn't really what you presented, but if you...
but it's fine. I personally don't think that it's the pathogen. Probably not. It's pretty much, it's quite well spread out. Like I said, go into GIS8, you will see that there's a lot of useful information in there. But it's a mix of maybe comorbidities, right? So it also depends on the comorbidities. I think the age as well. Of course, age then comes with other comorbidities. Unfortunately, it's sort of interlinked. Host genetics is definitely an area that many people are looking at. So there's a mix, it's a combination. So it's not really the pathogen, but it's a mix of the others that then will impact the immune responses, and that's what happens. Yeah, because if a patient has I think for comorbidities will actually impact the response as well. So I think it's a mix of factors.
Okay, great. I think we can stop there, Lisa. I want to thank you so much.
Thank you.
I didn't get the chance to introduce you, and there's so many nice comments about you in the chat. I just want to remind people that you've been voted the most inspiring woman at the Great Women of Our Time Awards for Science and Technology in 2005. You've been conferred the Junior Chamber International 10 Outstanding Young Persons of the World in Singapore 2013. And you've been conferred the highly prestigious Association of Southeastern Nations International Young Scientist and Technologist Award. And you're now the Executive Director of the A-Star Graduate Academy. Actually, you have so many more, and I won't go through all of them, but it's really just to let people know how privileged we were to have you with us today. There's so much feedback that people really loved your talk. So thanks so much for your time and for that really informative webinar.
Thank you, Faith. Thank you, Faith, for inviting me. I think it's good to see you again after 6 months or more, 8 months. I think. And I don't know when I'll get to see you again. Hopefully soon.
Hopefully soon, So I'd like to also thank all our attendees. Thank you so much for joining. The webinar will be available on demand in a few days' time. Thanks very much. Thank you for your questions, and we look forward to seeing you next time.
No, thank
Okay.
you. Thanks for everyone. Thank you. Bye.
Bye.